The overall goal of the following experiment is to demonstrate the role of external nucleating agents on inducing the freezing of plant material and to visualize the process using infrared thermography. This is achieved by lacing a drop of water and a drop of ice-nucleating active, or INA, bacteria on the surface of a leaf of the plant that is being studied. As a second step, the plant is placed into freezing temperatures and positioned so that it can be visualized with a high-resolution infrared camera. The parameters of the camera are then established, which will allow the researcher to observe ice initiation and propagation. Next, the temperature of the chamber is slowly lowered to sub-zero temperatures in order to induce the surface INA bacteria to freeze, which will then induce the plant to freeze. The results show the effect of extrinsic ice-nucleating agents to induce plants to freeze rather than supercool and avoid freezing, based on the ability of infrared thermography to determine the exact location of ice initiation and to visualize how ice is propagated throughout the plant.
This technique can not only be used to visualize plants under controlled conditions, but it can also be used out in the field under natural conditions. Understanding how plants freeze under natural conditions and getting information on the freezing process can provide new information that can lead to new techniques for frost protection of agricultural plants, but also tells us a lot about plant ecology, and this technique has been especially useful for understanding alpine ecology. Along with myself, Eric Burchard, a technician in my laboratory, will demonstrate this process in the following video.